Hello and welcome to this presentation on supporting the health of mitochondria. What we're covering today, we're covering the evolution of mitochondria, known as endosymbiosis, where two individual organisms join together through endosymbiosis to allow the formation of eukaryotic organisms. That's all complex life, which include organelles such as mitochondria. It's a really magical event that happened through cooperation between alpha proteobacteria and archaea. And archaea provided the host, and alpha proteobacteria evolved to become mitochondria. We'll also be looking at how mitochondria work, the importance of mitochondria and longevity, supplements to support mitochondria and health issues, the retrograde response, how mitochondria work with nuclear DNA to support longevity and mitochondrial health. So yes, a magical event happened, and it's only happened once in 4 billion years, an amazing event. And it's made all complex life and our very existence possible. This endosymbiosis, the ability for these two organisms to cooperate, and with alpha proteobacteria becoming mitochondria, providing the energy, the ATP that we need to keep ourselves alive, our longevity, and also the energy driving the evolution of all complex life. Everything you see in this picture, from animals, plants, fungi, all depend on this evolutionary step, this magical evolutionary step of producing mitochondria. So endosymbiosis, as I said, it's the evolution of alpha proteobacteria. They were an aerobic organism combining with archaea. Archaea provided the host and the food and the nourishment, and alpha proteobacteria became the organelle within this eukaryotic organism that evolved to build a building block of all complex life. And endosymbiosis, it means becoming by living together. We kind of almost taught that evolution is this competitive event, survival of the fittest, but no, this was a magical act of cooperation. Endosymbiosis have given eukaryotes, these are the, as I said, they're the building blocks of all animals, plants, and fungi. And it's given them thousands more times ATP per gene compared to bacteria and archaea, the individual organisms that they've come from, that those organisms cannot evolve because they don't have sufficient ATP per gene. They cannot evolve to where we are now. We needed this magical event to provide all the ATP per gene to support the number of proteins required for complex life. We're also powered by trillions of bolts of lightning every single moment of our lives. And that sounds too fantastic to be true, doesn't it? But if you think about the mitochondrial membranes, how wide those membranes are, they're very narrow. They're 6 nanometers. And it's 150 millivolts across that membrane. And if you expand that field strength to a meter, that is 30 million volts per meter, or bolt of lightning. So we've got millions and trillions of little bolts of lightning across our mitochondria, giving us our energy and ATP every single day of our lives. This important work on understanding how much energy mitochondria give us is from Nick Lane. Nick Lane is a really great academic who understands the evolution of mitochondria. He works from UCL. He's just really helped us understand how important mitochondria are. The name mitochondrion was coined in 1898 by biologist Carl Bender, from the Greek mitos, thread, and chondrion, little granule. An incredibly important woman also in understanding mitochondria is Lynn McGallis, and her seminal paper, The Origin of Mitosing Cells, really pushed forward the idea that mitochondria used to be bacteria. She was criticized quite heavily in the beginning, but she was so doggedly determined. She so believed this was to be the case, and she was right, and I love her for it. She was right. She proved a whole load of academics wrong. And it's actually the discovery of mitochondrial DNA proved that she was right. Proved that she was absolutely right. And Richard Dawkins also admires Lamberts too, and he admires her sheer courage and stamina by sticking to the endosymbiosis theory and carrying it through from being unorthodoxy and being criticized to an orthodoxy. And he's referring to a theory that the eukaryotic cell is a symbiotic union of primitive prokaryotic cells, that's alpha proteobacteria and archaea. And this is one of the great achievements of 20th century evolutionary biology. And yeah, she's admired so much now, but it took her several decades or more to prove her case. And eventually, as I said, with the discovery of mitochondrial DNA, she was proved right. So we have 4,000 trillion mitochondria in our bodies. And it's these will power our bodies and make sufficient ATP to keep us alive. And even more, another stunning figure, if we laid all our mitochondria out flat, these 4,000 trillion mitochondria flat, it would actually equate to 4 football pitches. So imagine 4 Wembley stadia, the football pitches on 4 Wembley stadia. If they laid out for one person, that would be all the membranes and our mitochondria are giving us ATP. Mitochondria are also effectively solar powered. The food we eat through photosynthesis, we're eating the food either from animals or from plants that through photosynthesis have picked up photons of sunlight and transferred that energy into food. And then when we eat that food, those high energy electrons that have been produced by the photons of sunlight are released into the electron transport chain, and mitochondria harvest this sunlight energy. We don't really consider that much, but we're solar powered. We're eating food that either, as I said, animals or plants that through photosynthesis are carrying that energy, and that energy is released into our electron transport chain. The heart is the most energetically demanding organ of the human body. Heart mitochondria turn over 6 kilograms of ATP each day to meet the requirements of contraction. It's roughly 15 times the weight of the heart. Each cardiomyocyte contains between 5,000 and 8,000 mitochondria. The liver represents, liver mitochondria represent 1/5 the volume of hepatocytes. So the liver is very hungry for ATP too, considering it's a chemical factory. It has to process so much in a day. So this can be 500 to 4,000 mitochondria per cell involved in several key metabolic networks and signaling pathways. The human brain, individual neurons, say of the substantia nigra, the dopamine producing neurons, have about 4.5 meters of axons. And similar cholinergic neurons predicted that they can have 100 meters of axons arising from a single cell body. They're so incredibly complex neurons, but they also need a lot of mitochondria. So in the substantia nigra, if you think it could be 100 to a few thousand mitochondria in the heart and the liver, but it could be 2 million in one neuron of the substantia nigra to fuel these axons, dendrites within these neurons. And if you take that same sort of concentration, it could mean there's as many as 40 million mitochondria in a cholinergic neuron. Yeah, so from a few hundred to a few thousand in the average cell, but in the brain, it can be several million. No wonder we get so tired if our energy levels get low, because our neurons are so hungry for mitochondrial ATP. How mitochondria work? So mitochondria, they can take glucose through the tricarboxylic acid cycle, so glucose through glycolysis, and they can, through oxidative phosphorylation, make energy. There's also beta oxidation of fatty acids as well. And another pathway is amino acids, where glutamine and glutamate can be taken into mitochondria and used for ATP as well. Interestingly, taking pyruvate from glycolysis into mitochondria requires several steps and several B vitamins, and this diagram shows it quite clearly. The first step, so pyruvate has to be activated by thiamine pyrophosphate, TPP, so that's B1, and TPP also needs alpha lipoic acid. Also, the lipoic acid needs to be activated by vitamin B2, FAD, flavin adenine dinucleotide, and also NADH as well is needed, and also B5. So coenzyme A is dependent on B5. So the whole process of getting pyruvate into mitochondria requires B1, B2, B3, lipoic acid, and B5 as well, the whole range of B vitamins. In the first step of energy production within mitochondria, this involves the Krebs cycle or the tricarboxylic acid cycle. And this provides the NAD and the NADH to go to the electron transport chain and some ATP as well. The Krebs cycle or the tricarboxylic acid cycle, this diagram, if you look where the blue arrow is, it's at the heart of all our metabolic pathways. You look at these complex diagrams, these complex metabolic pathways, at the heart is the Krebs cycle. So just how important it is, right at the heart of all our metabolic pathways. And again, Nick Lane, that wonderful Nick Lane, has described it as like the Swindon, the Magic Roundabout in Swindon, where all the vehicles come in and they leave in a really complex way, confusing way as well. The electron transport chain is high energy electrons, which are produced from sunlight through photosynthesis, are bound into NADH, and this NADH releases these high energy electrons into the electron transport chain, into complex 1 to 4 of the electron transport chain, and used to pump protons into the intermembrane space, and then those protons are then released and pushed through ATP synthase. And it's a bit like a hydroelectric dam, actually, that the energy from the intermembrane space drives a turbine-like protein ATP synthase, and this produces our ATP. Mitochondria and longevity. Unhealthy mitochondria can accelerate aging. Mitochondria are considered to be the main source of reactive oxygen species in our cell. And oxidative stress within mitochondria leads to damage to and stress in our mitochondrial DNA, which leads to reduced energy and ATP. And mitochondrial dysfunction also can lead to apoptosis or programmed cell death. Mitochondria, if we look after them, can protect us and give us energy, but mitochondria also will drive apoptosis, which is something which is linked to the loss of neurons in Alzheimer's, Parkinson's, and many inflammatory diseases, and also loss of energy and something like multiple sclerosis with the myelin sheath suffering through lack of energy. Mitochondrial quality and aging. Mitochondria have a life cycle, and they have to be fused together. They have to split apart all the time, and this does not happen sufficiently in neurodegeneration, but all degenerative diseases don't have sufficient mitochondrial quality control, giving us less and less energy. So mitochondria, they have this dynamic mechanism where they fuse together and make larger mitochondria, and then they split apart through fission. And then in fission, they're then tested to see whether they're up to making sufficient ATP. And if they're not, they're digested through a process called mitophagy. That's the self-eating of mitochondria. So it's a continual quality control process. And interestingly, insulin resistance, obesity, type 2 diabetes will undermine this process. Also, mitochondrial biogenesis occurs in this cycle too. We make more mitochondria in this cycle as well. Yeah, so mitochondrial autophagy or mitophagy is the house cleaning that's needed to eat our own mitochondria. And long-living species need to have this process, like humans are usually really good at digesting our worn-out mitochondria to help maintain their quality and keep ATP levels up. And as I mentioned, insulin resistance, hyperglycemia can undermine this dynamic process, leading to dysfunctional mitochondria, increasing the risk of type 2 diabetes and Alzheimer's disease as well. So to initiate mitophagy, to make sure we're clearing out our worn mitochondria, there's several nutrients such as genistein, hops, quercetin, curcumin, sulforaphane from cruciferous vegetables, tocotrienols, vitamin E, vitamin C, and selenium are all really good at initiating mitophagy. So what helps us make more mitochondria through mitochondrial biogenesis? Well, making sure we voluntarily cut down on our energy input. Now, whether that's through low energy, through exercise, intermittent fasting, this will activate enzymes such as AMPK, and the anti-aging proteins such as SIRT1 will send signals to a protein within our cells called PGC1 alpha, and this triggers our mitochondria to undergo mitochondrial biogenesis. Conversely, the typical Western diet with excessive calories will undermine this process. Quite often you see it in studies called overnutrition, which is not really, it's a bit of a misnomer, but it's just highly calorific diets will undermine mitochondrial biogenesis, whereas exercise, intermittent fasting will lower the levels of energy, ATP and NADH, which will trigger these enzymes to make more mitochondria. The body's sensing this lower levels of energy and then has to make more mitochondria to replace those levels of energy. So yes, quercetin, genistein, resveratrol, anthocyanidins can also help us make more mitochondria through mitochondrial biogenesis. And it's this important protein called PGC1 alpha, which is supported to make more mitochondria. We do need low levels of reactive oxygen species generated by exercise and plant polyphenols, surprisingly, to protect mitochondria. So small amounts of reactive oxygen species. If you think polyphenols are plants protectors, they're designed to protect from various pests, and they do have they do produce a small amount of reactive oxygen species, as well as being really good antioxidants too, but they do produce small amounts of reactive oxygen species, which will, if you see in this diagram here, that something like sulforaphane, curcumin, and flavonoids produce small amounts of reactive oxygen species, which will precondition our mitochondria to be able to withstand higher levels of reactive oxygen species. And this is known as hormesis. And species such as humans, they excel at this hormesis. And the hormesis also includes communication with mitochondria with our nuclear DNA as well. So long-lived species do this really, really well. If we look after our mitochondria, we engage in things like exercise, plant polyphenols, they can support our mitochondria to engage in this hormesis to protect our energy levels long-term. So low levels of reactive oxygen species generated by exercise protect mitochondria too, in a similar way, this hormesis. In exercise, we know exercise generates reactive oxygen species, but our mitochondria respond really well to this by becoming stronger and producing more ATP. In fact, someone that's exercising, an athlete that's exercising, is not just practicing their discipline, they're also making more mitochondria so they can excel in competition. Supplements for mitochondria. Carnitine. Dietary long-chain fatty acids require the amino acid carnitine along with CoA, so vitamin B5, to transport these fatty acids into mitochondria. Once inside mitochondria, fatty acids undergo beta oxidation, the first step in providing energy from fatty acids. And in this diagram, it shows you these steps. So we need carnitine to get long-chain fatty acids into mitochondria, and this requires B5 for the CoA. Also, acetyl L-carnitine is a good nutrient to support mitochondria too. So mitochondria, they have a glue, a phospholipid glue, which holds all the complexes in place, and it's called cardiolipin. It's a really important phospholipid to hold the integrity of the inner mitochondrial membrane together. And acetyl L-carnitine can support the synthesis of cardiolipin, which decreases with age. And this can not only improve fatty acid transport into mitochondria, but has the remarkable ability to maintain the structure and function of the inner mitochondrial membrane. A really important part of mitochondria, which holds all these complexes which are necessary to pump protons into the intermembrane space and allow these protons to come back through ATP synthase to make ATP. As I showed you earlier as well, all the B vitamins necessary. So B1, lipoic acid, B5, B2, and B3, all important to keep mitochondria functioning. Vitamin B3. Vitamin B3 is a precursor to the coenzymes nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, which are needed to enhance mitochondrial activities for the synthesis of ATP. So vitamin B3 is really important. Coenzyme Q10. The conversion of energy from carbohydrate and fat to ATP requires the presence of coenzyme Q10 within the inner mitochondrial membrane. So yeah, it's involved in the shuttle of electrons through the electron transport chain, and it's a vital component of mitochondrial energy production. Interestingly, sometimes coenzyme Q10 is difficult to absorb through the gut, and interestingly, vitamin K, being a quinone like CoQ10, which is ubiquinone, ubiquinol. Vitamin K is a quinone in the same family as CoQ10 and can stand in some circumstances, combined with vitamin C to support the electrons transport in the inner mitochondrial membrane. Magnesium. Magnesium deficiency leads to reduced mitochondrial energy and increased oxidative stress. Both low ATP and high reactive oxygen species are significant factors in mitochondrial dysfunction, and magnesium can make sure that we help maintain our ATP levels and reduce our reactive oxygen species levels. And if it reduces, then it does affect our energy production. Interestingly, many ATP molecules are supported in structure by magnesium, by a molecule of magnesium. Taurine. The amino acid taurine was originally isolated from bull semen, hence its name Taurus, after the bull. Within mitochondria, taurine can release energy by supporting ATP synthesis, decrease oxidative stress, decrease inflammation, and protect mitochondria from calcium overload. Excessive amounts of calcium entering into mitochondria can lead to apoptosis. Within neurons, calcium can drive mitochondria and lead to excitotoxicity because mitochondria are sensitive to calcium and try to make more ATP, but excessive calcium can overdrive mitochondria beyond the point at which they can make sufficient ATP and lead to their death. So by making sure we have sufficient taurine, can protect mitochondria from this excessive amount of calcium and buffer the calcium to maintain ATP levels. Resveratrol. Resveratrol activates anti-aging genes which switch on the proteins needed to make new mitochondria, so mitochondrial biogenesis. So the SERT proteins can activate mitochondrial biogenesis, you can see in this diagram here. Also, resveratrol supports PTC1 alpha, which I keep talking about. It's needed for mitochondrial biogenesis. Glutathione. Glutathione, it's an antioxidant. It's vital for the correct functioning of mitochondria. 10% of cellular glutathione is imported into mitochondria. Any less than this will lead to oxidative stress and energy failure and drops in ATP and increases in the risk of apoptosis too. The top diagram shows you that 10% is needed, 10% from the cell, from the cytosol is needed to maintain mitochondrial function. And if this drops below 10%, this can lead to energy failure and apoptosis and cell death. Korean ginseng. Korean ginseng compounds target mitochondrial reactive oxygen species. They protect against apoptosis. They support mitochondrial energetics, support mitochondrial biogenesis, support the dynamics, the making sure the quality control of mitochondria is carried out as through mitophagy. Reishi mushroom. Results of an animal study revealed that reishi mushrooms significantly advance the activity of mitochondrial Krebs cycle, or the tricarboxylic acid cycle, and the electron transport chain complexes. And reishi mushroom upregulates heart glutathione peroxidase, glutathione transferase, catalase, superoxide dismutase, and reduce glutathione as well. So it does some pretty good things to support mitochondria. It also decreases the highly toxic radical hydroxyl radical, and also the membrane destructive lipid peroxide malonaldehyde, which will damage mitochondrial membranes too. Vagal stimulation. The vagal system plays an important role in maintaining mitochondrial homeostasis and suppressing mitochondrial oxidative stress. Mitochondrial contact site, cristae organizing system, which sounds a bit complex, but MICOS in the mitochondrial membrane is crucial for the formation and maintenance of this cristae in a mitochondrial membrane structure. And vagal stimulation is really important for maintaining this structure. And the neurotransmitter acetylcholine supports this whole function. And there's receptors for acetylcholine in mitochondria to support this function. And vagal tone can be stimulated by a variety of activities. Being outside in green country spaces, called a green prescription by some practitioners, some medical practitioners. Social connection, being around good people. The vagus nerve can trigger the production of oxytocin, which will really help with mitochondrial and brain function. Singing, music, laughter, crying, yoga and meditation, exercise can all support this vagal connection, acetylcholine synthesis to protect mitochondria. Mitochondria health issues. So mitochondria, insulin resistance is something that really undermine mitochondria and mitochondria energetics, leading to something called lipotoxicity, which damages mitochondria. Insulin resistance is something that's really, really damaging to mitochondria. And this will affect skeletal muscle, liver, brain, adipose tissue, pancreas, and the blood too. Insulin secretion is mitochondria dependent. So the pancreatic beta cells sense glucose coming into the pancreatic beta cells, and mitochondria respond by making more ATP, leading to the secretion of insulin. So it's dependent on mitochondria. Fatigue. Fatigue is a hallmark symptom of mitochondrial dysfunction. It's often described by patients as a lack of energy, mental, physical tiredness, diminished endurance, and the need for a prolonged recovery after physical activity. Sometimes it's difficult to rest enough with this kind of fatigue and mitochondrial dysfunction. In the diagram, we can see that damaged mitochondria, poor functioning of the electron complexes in the electron transport chain, oxidative stress, inflammation, impaired fatty acid metabolism. In fact, that's one of the first things to go, is the fatty acid metabolism can sometimes struggle, particularly in MS. We see this in MS, that people struggle with their fatty acid metabolism. But eventually, with poor mitochondrial function, will lead to mitochondrial dysfunction, inflammation, and increased fatigue. Neurodegeneration. Neurons are incredibly hungry for ATP energy, and the neurons have a chauffeur. They're called kinesin proteins, and these proteins chauffeur mitochondria along the synapse up along the axon up to synapses to deliver ATP energy. But synapses will be incredibly hungry for ATP energy. Not all mitochondria are delivered in this way as cargo with these kinesin motors. Some remain local to the synapse, but many are delivered. And it's a really important process that goes wrong within neurodegeneration. So there's these structures within our neurons called microtubules, which give our neurons its shape, its form and shape. That this unusual shape of a neuron doesn't happen just on its own. It has to have a cytoskeleton, and this cytoskeleton is used to deliver mitochondria up and down, also take waste products away as well. And this does not work sufficiently in Parkinson's and Alzheimer's, and particularly in Alzheimer's, the cytoskeleton starts to collapse, which means that mitochondria cannot be delivered. And insulin resistance is one of the big drivers in the loss of these microtubules. Depression, mitochondrial dysfunction occurs in depression. So there's less ATP being produced by mitochondria in the brains of people with depression. So it could be mitochondrial DNA mutations, meaning they can't make sufficient components of mitochondria. The membranes become more permeable, meaning that they don't function that well, allowing too much calcium in, increasing reactive oxygen species. So yes, in depression, we do need to support people's mitochondria. Hypertension, the ability to vasodilate, requires mitochondria to be functioning to enable vasodilation in the endothelium of blood vessels. And excessive oxidative stress can undermine these mitochondria, leading to impaired vasodilation. Healthy mitochondria are needed for heart contraction and relaxation. And so if there's not sufficient ATP, mitochondria cannot allow the sufficient relaxation of heart muscle and contraction of heart muscle. Heart disease. Heart disease, there's some new ideas coming out about heart disease and heart failure, because many of the drugs used in heart disease are designed to reduce the energetic demands of the heart. And the newer philosophy is, well, if you've got mitochondrial dysfunction in the heart, in heart disease, why not support mitochondria to produce more energy so that this dysfunction, mitochondrial dysfunction, can support energy production in the heart and help repair the heart and maybe make it less likely that the drugs are needed in heart disease. So rather than produce the energy demands of the heart through drugs, well, how about supporting mitochondria in the heart so that the energetic demands can be met more easily? There's an interesting story in America. Georgia Bowen, she had a heart attack within the womb and struggling to survive. So Georgia Bowen had this heart attack, and the doctors from the Boston Children's Hospital decided that they would try an experimental procedure, and they took a billion mitochondria from a small plug of Georgia's healthy muscle and fused them into the injured heart muscle and helped her survive. It's an amazing story. Mitochondria and osteoarthritis. Chondrocytes, we think about osteoarthritis as being wear and tear, but the chondrocytes within cartilage, they need mitochondrial energy to maintain their integrity to protect against osteoarthritis. In the diagram on the right-hand side, you can see all the things we've been talking about maintaining healthy mitochondria, fusion, fission, biogenesis, making sure that there's sufficient mitophagy to clear out worn-out mitochondria. So mitochondrial energy within our chondrocytes can help protect us against osteoarthritis. In bone, bone needs functional mitochondria to be able to build up bone and resorb bone too. So Osteoblasts, they build up bone. Osteoclasts, they resorb bone to help us repair bone as well. The thing with osteoporosis is there's insufficient osteoblast activity and excessive osteoclast activity. One of the frustrating things with osteoporosis is that an inflammation is that we tend to get, with mitochondrial dysfunction, we tend to get less osteoblast activity and excessive osteoclast activity. So there's the inability to build up the bone, but accelerated degradation and resorption of bone as well. So by supporting our mitochondria, we can protect against osteoporosis as well. Mitochondria in ovarian aging and reproductive longevity. So ovarian aging can be accelerated by mitochondrial dysfunction, leading to infertility in females, but also the same can happen in semen too, that the mitochondrial dysfunction within semen can also undermine fertility in males. Because many of the mitochondria, in fact, most of the mitochondria within semen are situated in the tail of semen, leading to male infertility. The retrograde response, I talked about the hormesis response, the ability to there's a little bit of toxicity which can lead mitochondria to produce protective mechanisms to maintain longevity and the health of mitochondria. So when mitochondria produce some reactive oxygen species, they initiate something called the retrograde response, which asks mitochondria for help to make synthesized proteins that support mitochondria. And humans do this really, really well. Humans have a good conversation between mitochondria and our nuclear DNA. So when under stress, this signal goes to our DNA, and the DNA respond in kind by making mitochondrial proteins and also protective antioxidants as well. So this the important communication between mitochondria and nuclear DNA requires nucleotides and nucleosides. And these are needed to help our mitochondria talk to DNA. So we don't often talk about nucleotides and nucleosides. And we can make them from fresh. We can get them from the diet. Or we can salvage them from food as well. And I've got this this picture here to show the salvage pathway to indicate that it's important to salvage these nucleotides and nucleosides to make use of them again, so that our mitochondria can communicate with our DNA. So yes, the salvage pathway or de novo synthesis of making them from fresh. And it's a highly energetic process to make these nucleotides and nucleosides from fresh. So it's a really good idea to salvage them through the purine pathways and the pyrimidine pathways to make sure that we're not using up too much energy to make them from fresh. But if we salvage them and reuse them, this enables mitochondria to talk more clearly with our DNA. So the degradation products of RNA and DNA can be salvaged to make nucleotides and nucleosides, which allow retrograde communication between mitochondria and nuclear DNA. I just re-emphasize that point, that these nucleotides and nucleosides are really important to salvage them to be able to communicate between mitochondria and DNA to maintain our mitochondrial health throughout our lives. The synthesis of these from these de novo synthesis also requires folic acid as well. Defective mitochondria in Parkinson's models send SOS signals to the cell nucleus and involves a demand increasing the synthesis of their own genetic material. They use a major pathway called the salvage pathway, which I keep repeating, for mitochondrial DNA synthesis to do this. Data support the therapeutic potential of folic acid to enhance nucleotide pools, promoting mitochondrial biogenesis, improving mitochondrial function in neurons, and protecting against neurodegenerative disease as well. And think about just ATP itself. Adenosine, where does the adenosine come from? NADH. These nucleotides and nucleosides are needed in the ability to carry energy as well, not just in this this pathway, not just this retrograde response. They kind of undervalued some of these nutrients, and it's particularly important for the function of mitochondria. Also, the de novo synthesis of purines, pyrimidines, these nucleotides requires mitochondria to be able to make some of these components too. So yeah, folate is important in this pathway as well. Diet and mitochondria. Mediterranean diet. Mediterranean diet is associated with lower oxidative stress levels and a lower incidence of chronic disease. Compounds within the foods contained in the Mediterranean diet can improve mitochondrial function and also reduce cardiovascular and metabolic risk, promoting health and longevity. I mean, the Mediterranean diet works for so many different health conditions, but it's particularly important for mitochondria, containing chlorogenic acid, delphinidin, lycopene, resveratrol, polyunsaturated fatty acids, polyphenols, particularly rich in extra virgin olive oil. Other bioactive compounds improve oxygen consumption, upregulate mitochondrial activity and ATP synthesis. So genistein, quercetin from onions, apples, anthocyanins from grapes and berries, cinnamon, chlorogenic acid, which is also quite rich in coffee as well, and resveratrol, obviously from grapes. Whole grains. Natural bioactive components within whole grains can help protect against metabolic disorders by modulating mitochondrial genesis, biogenesis, and energy metabolism. So there's many different components in whole grains which are supportive for mitochondrial function. Ketogenic diet. Ketone bodies such as acetoacetate and beta-hydroxybutyrate lead to decreases in oxidative stress and improvements in mitochondrial biogenesis and mitochondrial function. Ketogenic diets can be supportive too. Sometimes people, if they've got mitochondrial dysfunction, that a high-fat diet may overwhelm beta oxidation. So some people have to be careful. But in general, the ketogenic diet, particularly if it's a ketogenic diet that's not too stressful on metabolism, can make ketones which are protective for mitochondria. Tests. Organic acid test. Many organic acid tests have components or markers for the Krebs cycle or tricarboxylic acid cycle, and also some markers for the function of the electron transport chain as well, particularly CoQ10. There's a mitochondrial profile available from the Academy of Nutritional Medicine. I've got their email there. If you're interested in finding more about that, I've got their email there if you want to contact them. It gives you a lot of information about the capacity of mitochondria to make ATP, mitochondrial DNA changes, also the levels of activity of PGC1 alpha, that protein that I keep mentioning for mitochondrial biogenesis. Genetic profiles such as LifeCode GX, they test for polymorphisms that look at the polymorphisms in various enzymes which affect the Krebs cycle in particular, and mitochondrial function in particular as well. So I love this statement here. If we wish to live longer and to rid ourselves of the diseases of old age, we'll need more mitochondria, and healthy mitochondria, of course. So what we've done today, we've looked at how mitochondria provided the energy that allows evolution and the very existence of all complex life. Mitochondria provide the ATP per gene to allow organisms to experiment with really complex proteins to be able to adapt to their environment and excel, thrive, and evolve in their environment. That requires mitochondrial ATP. The charge across the mitochondrial membrane is equivalent to a bolt of lightning. That's the work of Nick Lane. Just a few nanometers, 150 millivolts across a few nanometers equates to several million volts per meter. And that's absolutely stunning, just how much energy our mitochondria produce in our trillions of mitochondria. Mitochondria are effectively solar powered. We harvest the high energy electrons that are captured in our food because of photosynthesis and because the animals eating the plants themselves. We release those high energy electrons into our electron transport chain to give us the ATP we need to fuel our lives, to power our lives. Mitochondria digest themselves every few days to maintain their quality and integrity. They have to, otherwise they become dysfunctional automatically, and we have to make sure we get rid of the ones that cannot make sufficient ATP because they're one of the main sources of reactive oxygen species in the cell. We do not want mitochondria producing too many reactive oxygen species. Hyperglycemia and insulin resistance lead to mitochondrial dysfunction. Insulin secretion is dependent on beta cell mitochondria, that glucose entering into beta cells activates mitochondria to make more ATP to trigger the release of insulin from the pancreatic beta cells. Fatigue is a hallmark symptom of mitochondrial dysfunction, that our energy levels will drop through the inflammation and insufficient energy being made by mitochondria, and it's a hallmark of fatigue. Bone and cartilage depend on mitochondrial energy for their integrity. That bone, insufficient mitochondrial function in osteoblasts will lead to poor bone formation, but will also lead to the increased resorption of bone, accelerating the risk of osteoporosis. Cartilage also needs to have sufficient mitochondrial function to power the chondrocytes to protect ourselves from osteoarthritis. The heart is the most energetic demanding organ of the human body and requires over 6 kilograms of mitochondrial ATP per day. That's kind of ADP, ATP, ADP, AMP. It's the backs and forwards. It's the processing of that molecule of the ATP. That means there's 6 kilograms per day being used by the heart. So thank you. Thank you very much. I hope the lecture was informative.